Okay, this part of A&P talks about the cell, and most of this again may be a review to you. We're going to go over the different organelles inside the cell. We're going to also talk about mitosis. We'll talk about cell death, and we'll even talk about transport, active and passive transport. So, starting with um, the very beginning, we know what a cell is. As we went over the levels of organization, we know that the cell is the basic unit of life, and we have many different types of cells. And the reason why we call them differentiated is because they have various shapes, sizes, and functions. And this slide here kind of shows us the wide variety of cells that we have in the body. For instance, our blood cells are here, red blood cells and white blood cells that are drastically different from just normal body cells. And that shape is drastically different from neurons, and that is drastically different from muscle cells. And even muscle cells themselves can be different from each other, whereas this is a smooth muscle cell and this is a cardiac muscle cell, and this is an epithelial cell. So all cells are going to have a little bit of difference among them, and but they will all have some of the basic things alike. For instance, all of them will have a nucleus, cytoplasm, cell membrane. And we'll go over all of the different organisms inside the cell, but generally speaking, we know that the nucleus is the control center of the cell. It's going to be where genetic information is held. The cytoplasm, usually people think of it as just being that fluid that all the organelles are in, but the cytoplasm is actually the fluid plus the organelles. If we're just talking about the fluid or the liquid, that's going to be the cytosol. But if you include the organelles, now we have the cytoplasm. And then our cell membrane, which will be one of the first organelles we'll talk about, is what surrounds the cell and is going to allow things to enter and leave. And again, this is just a general structure of a normal animal cell, and we will go over the different parts of it as we move forth through this chapter. So, the cell membrane. Cell membrane, as I said, is going to be responsible for allowing things to go in and out of the cell. It's going to be what we call selectively permeable. And permeable simply means that it allows certain things or allows things in and out, but because it is selective, it only allows certain things in and certain things out. So it's the regulator to say, oh, this thing we can't just let you pass through. We may have to have some type of transport protein. We may need a little bit of help or you can't get in at all. And we also have what we call signal transduction. And that just means that signals or things on the outside can allow or cause signals to be sent that may allow certain protein channels to open up for some type of facilitated diffusion to happen so the cell can respond to messages that may come to it. And the cell membrane is going to not just be this simple lipid bilayer, but this is just the phospholipid bilayer that you may have seen before. So we have two layers of phospholipids. But it's not that simple where we have the phospholipids all the way around. We also have proteins and carbohydrates that are embedded into the cell membrane. And this is what I was just referring to, that phospholipid bilayer, where those hydrophobic and hydrophilic parts, the hydrophilic part, that's going to be the water loving. And so that's one part of it. And then the hydrophobic is the water fearing. and that's the hydrophobic tails and then the hydrophilic heads. Okay, and so the bilayer is primarily going to be permeable, which means that it's going to allow certain things in to those that are of lipids, but impermeable to water soluble substances. So those that are water soluble, they may need to have some type of carrier protein to allow them to come in. And again, we said we have a lot of different things inside of the cell membrane, like cholesterol, and we'll talk about that a little bit later. Also, lipids and proteins, because they're inside the cell membrane, they have a bit of flexibility, so the cell membrane is not as stiff as, say, in plants. And so it has this fluid mosaic property, the cell membrane itself. And we also have, again, a lot of proteins that may be receptors, it may be channels that are going to allow things in and out. And they also help to induce signal transduction, as we mentioned on the last slide. So this is an actual picture of what the cell membrane will look like. And so this part here is that phospholipid bilayer that I was attempting to draw. But all these other structures, you have your glycoproteins, your glycolipids, we have peripheral proteins here, cholesterol molecules that you can maybe see those small structures there. So the cell membrane is not just a phospholipid bilayer, but there are many other structures that are embedded inside. Next, we have our cytoplasm. And as I said before, the cytoplasm is not only that fluid or that liquid that the organelles are inside, but it is the organelles and that fluid. So the cytosol itself is the fluid, but if we have that plus the organelles, now we have our cytoplasm. Okay, and we're going to talk about all the organelles that are inside of that. And also within the cytoplasm, we have what we call a cytoskeleton. We'll talk about what makes up the cytoskeleton, but it's there to give it a bit of structure and a little bit of a framework so that things can move about the cell as needed. All right, our next two organelles, ribosomes and the ER. Now they are together because they kind of work together. So we have our ribosomes. Our ribosomes are going to be responsible for protein synthesis, and this is something that is talked about later, but that's the primary function of our ribosomes. When we go into protein synthesis, we'll bring up our ribosomes then. Our ER, now we do have two different types of ER. We have rough ER and we have smooth ER. Rough ER is called rough because it contains ribosomes. And because it contains ribosomes, it's going to also be responsible for protein synthesis. Smooth ER does not have ribosomes, and that's why we call it smooth. And it's responsible for making our lipids. Okay? And also, once these things are made, the ER is going to be responsible for transporting those. So this is our transportation system. So this is what's going to be responsible for taking things where they need to go and sending them out, making those vesicles so that they can be sent out. Next, the vesicles. And so after those things are made at the ER, or maybe they're made at the smooth ER or rough ER, whether it is a protein or a lipid, they form vesicles, which are just sacs of material. And they're going to be responsible for taking that material and transporting it where they need to go. And then our Golgi. Golgi apparatus, I like to kind of think about as being our UPS. Our UPS is going to be responsible for packaging and shipping things where they need to go. So those things that are made on the rough ER, the Golgi is going to be responsible for packaging those things up and shipping them out. And an example of how organelles interact with vesicles is milk secretion, whereas milk is produced, and when the milk is produced or the proteins that are needed within milk, vesicles then are going to package that milk and maybe even the milk proteins, and they will be secreted as vesicles or by vesicles. Next, the mitochondria. Now, the mitochondria, we usually hear powerhouse of the cell. But the key word in this is power. The power is ATP. This is that energy that is created. And it's created, this energy is created through cellular respiration. So the energy is ATP, created through cellular respiration, and this happens within the mitochondria. Next, our lysosomes. Like to think of the lysosomes as being the garbage disposal, which means that they're getting rid of any type of waste, anything that needs to be disposed of, whether it is a cell that's no longer working or cell parts that are no longer working, anything that needs to be digested, bacteria that may need to be digested. That's going to be what our lysosomes are going to do. Peroxisomes are going to do the same thing as lysosomes, but the only difference is that our peroxisomes are not going to be digesting proteins and carbohydrates, but in this case, they're going to be digesting lipids and alcohols. And so, same exact function, they just digest two different things. And then we have our centrosome. Now, centrosome is going to be the collective term for the two centrioles. And you may remember centrioles from when you learned about mitosis, where with mitosis, towards the beginning of prophase, the centrioles will move to opposite poles of the cell and help to form spindle fibers. And so they are going to show up a little bit later as we go through mitosis. Cilia. We talk about cilia. It also usually kind of talk about flagella. And cilia and flagella are similar in that they help with movement. It's going to be responsible for movement. That's why it says modal extension. So movement. Moving things where they need to go. For instance, to propel mucus in the respiratory tract. Whenever mucus collects or surrounds any type of dirt or debris, we want to get that out of the respiratory tract. And so we have cilia on the cells inside the respiratory tract. Also, when the egg from the female is released from the ovary, we have cilia that's going to push that egg towards the fallopian tube and also towards the uterus after it has been fertilized. These are more like they're hair-like shortened structures that help with movement. And then we have our flagella. With our flagella, you usually are going to have only one, whereas with cilia, you may have plenty and many of them for a single cell. And the only flagellum for a human cell is going to be the tail of a sperm cell. And that's probably the most familiar one that you think of when you think of a flagellum. But it's also for movement. And then we have our microfilaments, microtubules, and intermediate filaments. Now, if you all remember a few slides ago, I mentioned something about the cytoskeleton being a part of the cytoplasma to help give it a structure and kind of help support it. And so that's what these three are going to be responsible for, especially the microtubules and intermediate filaments. They're going to be responsible for that shape and that structure. Okay, and they also help move things around. For instance, during mitosis, microtubules will help move those structures around like they need to. Microfilaments will be something that you'll hear more about when you talk about the muscular system, because it helps to make up actin. And actin is one of the microfilaments that is needed for muscle contraction. And we have these again in our cells. All right, so cell nucleus. This is one that we mentioned earlier, where we know that the nucleus is going to be our control center of the cell. It's going to have our genetic material, i.e. the DNA. And in order to get inside of the nucleus, you have to have some type of pore or a membrane. And so we have an envelope, just like we have a cell membrane around the cell. We have somewhat a nuclear membrane or nuclear envelope that allows things in and out of the nucleus. And so it allows certain substances to come in and out of the nucleus. And so here is where you'll see our nuclear pores, those little small structures there, those openings within it that allow things in and out. And then our nucleolus, found in the center of the nucleus, and this is where you will find our ribosomes being produced. So ribosomes are produced in the nucleolus. They will then have to leave eventually, and they exit via the nuclear envelope or the nuclear pores within the nuclear envelope. And as we said before, the nucleus is going to have genetic material, but it is not always in the form of a chromosome, and so it's mostly going to be in the form of chromatin, which is just DNA that is kind of stretched. It's around proteins, but not really condensed like a chromosome. All right, so this next section for this chapter will talk about passive and active transport. How do we get things in and out of the cell? How do we allow things to go in and out of this selectively permeable cell membrane? And so the first few we're going to talk about will be passive transport. And passive transport means no energy. So it's not require ATP, so it does not need energy. Whereas active transport does require energy. So you will have to have energy for this. You have to have energy, ATP. Okay? And we'll talk about each of these that are listed. So the very first one, again, these are all our passive transports. Diffusion. Diffusion is going to be where we have things moving from higher concentration to low concentration, and it will be different atoms, molecules, ions, things like carbon dioxide or any other substances, and it literally just goes from where we have high concentration. For instance, in this example, you have a high concentration of, let's say this is sugar. Well, it is sugar. Let's say this cube, this is a high concentration here. That means it's a low concentration of sugar in this area of the beaker. And so eventually, as it dissolves, it's going to go from areas of high concentration, and those particles are going to go into areas of low concentration until it has reached equilibrium. Another example that I like to use is, for instance, if I stood at the door of the classroom or close to the door of the classroom, and I sprayed some air freshener in the corner of the classroom. The first people that would probably smell that air freshener would be those that are closer to me, because that is where the concentration is the highest. So the concentration of that air freshener is going to be the highest in the corner where I first sprayed it. The concentration is going to be the lowest in the complete opposite corner. So farthest away, the opposite corner. But eventually, those particles of the air freshener will move from areas of high concentration, the corner where I first stood by the door, to areas of low concentration, which are the opposite corners of the room. And so it will continue to diffuse slowly until it reaches equilibrium, which means that it's basically the same throughout the entire room. This is just another example of particles moving from areas of high concentration to low concentration through a selectively permeable membrane. This partition here showing that membrane that is permeable to both water and some type of solute. That solute could be salt, sugar, and the solute is in high concentration over here, but lower concentration over here. Water is in high concentration here, but in lower concentration over here. And so, as time passes from one beaker to the next, you will notice that both the solute is moving from the left to the right, and the water is also moving from the right to the left, so from areas of high concentration to low concentration. And it continues again until the equilibrium is reached. And this is for with a membrane that is permeable to both of those substances. Another type of passive transport, which means that there's no ATP that is required, no energy required, is facilitated diffusion. Now, when you think about facilitate, so we know what diffusion is, but to facilitate means to kind of help it out. Because regular diffusion just happens on its own, but facilitated diffusion, there may need to be a helper. And so we have to have helpers or transporters or ion channels that are going to help certain substances cross the cell membrane that can't cross the cell membrane. Remember, earlier we said that the cell membrane does not allow water-soluble substances. And so in order for them to pass, we have channels that are embedded into the cell membrane that will allow them to cross from, again, regions of high concentration to regions of lower concentration with the help of carrier proteins. So from high concentration to low concentration with the help of a carrier protein. So these two will be our carrier proteins, whereas this is an ion channel, whereas it was just passed through. Now we have osmosis. Osmosis is still a type of passive transport, no ATP required. But in this case, specifically, we're talking about the passing of water. So the movement of water, and again, we're talking about from high concentration to low concentration. Okay? So from high concentration to low concentration, and this is where the movement of water is going from high concentration to low concentration. For instance, if we're looking at this beaker here, we have protein molecule that cannot cross the cell membrane, that selectively permeable membrane here. The blue is the water molecule that's here. And so high concentration of solutes are on this side. Low concentration of solutes are on this side. And so it's going to go, water is going to move so that it can be at equilibrium, it can be at a balance. It's going to go from, again, water is moving to a region of high solute concentration. So it's going to go from high concentration to low concentration. I'm sorry, it's going to move to a higher concentration so that it can reach equilibrium. So we need to have more water on this side to help balance it out. So water is going to move through the semi-permeable or selectively permeable membrane so that it can be at equilibrium. And now the solutes again did not move, but because we had more solutes on this side, water was able to diffuse across. And when water molecules are able to diffuse across, now both sides are at equilibrium. The concentration ratio is the same. Let's say this was salt. This would be just as salty as this side, because we have the same ratio of water and solutes now. All right, so the way that this is going to move, or the reason why it moves, is because of pressure, specifically osmotic pressure. And osmotic pressure is going to cause the movement of that water concentration from high to low. But where the solutes are the highest, that's where water is going to move towards. And we have three different types of solution that it can move in, or may not have much movement of water. The first one is isotonic. The prefix iso is going to mean same. And so for this one, we have the same amount of osmotic pressure on both sides, which means the solute concentration is basically the same on both sides. So there's really no net gain or loss of water. It's just going to kind of oscillate back and forth at the same rate. With a hypertonic solution, that's going to be the example A here with isotonic solution. A hypertonic solution is going to cause a solution to lose water. So remember, water is going to move from where water is in high concentration to low concentration. So that means that water is leaving out of the cell. So it's going to be leaving out of the cell, moving into a place that is high in solutes. So for instance, if I were to put a cucumber inside of vinegar, we have more water inside of the cucumber. We have more solutes inside the solution of vinegar. So in what direction would water move? Water is going to move from the cucumber, where water is very high, to the outside of the cucumber, where the vinegar is and water is very low. And eventually, it will shrivel. And so that is basically how we create pickles. And that will be our second picture here. So in a hypertonic solution, this is hypertonic here. This is iso. So iso, no movement or gain or loss of water. Hypertonic solution, that's the pickle example, where we have more water on the inside of the cell, more solutes on the outside. Water will move out. The cell will shrivel or shrink. And the last is a hypotonic solution. With a hypotonic solution, it's where we have more water on the outside of the cell. More water on the outside of the cell, more solutes on the inside, which means that water is going to rush into the cell. It's going to cause it to swell and then possibly even burst. Examples that I use or like to use is if you've ever taken a gummy bear, put a gummy bear inside of water, let it sit there for a while, it will eventually swell. Or any other liquids that you choose to put gummy bears in, but they will then swell. And that's a hypotonic solution because there's more water on the outside of the cell than there is on the inside. And so water will rush inside the cell, continue to rush until it swells and possibly even burst. Next, we have filtration. Again, filtration is a passive process, but in this case, those are going to be particles or molecules will be moved because of pressure that's being placed on them. And it could be various types of pressure, and you'll talk about that throughout different systems. But an example, for instance, when blood plasma leaves capillaries, we're going to have only certain things that are going to be filtered out. For instance, in the kidneys, the very first thing that happens is filtration. Tons of things are pushed out of the blood and into the nephron of the kidney, but large plasma proteins are not pushed through. So it's kind of like filter paper when you think about a filter of your car or a filter for coffee. When you're filtering coffee, many things can get through, but those grains are kept back. Those large things cannot get through. And again, this is still a passive process, no ATP. Next, this is going to be active transport. Again, with active transport, we do have ATP. And if you remember, for all the ones we just mentioned, we're going from areas of high concentration to areas of low concentration. From high concentration to low concentration. With active transport, it's going to be the opposite. We're going from areas of low concentration to areas of high concentration. So that means it's going against the concentration gradient, where we've been taught so far high to low, but now we're going from low to high. So we have to have a little bit of help, and of course, we have to have a little bit of energy in order for this to happen. So one example of active transport is endocytosis. Endo means inside, so that means we're taking some type of particle or substance inside of the cell. And endo means inside. And we have three different types of endocytosis. Penocytosis. Peno just means that we're engulfing a liquid. Again, all three of these are in reference to taking something inside. Phagocytosis, engulfing a solid. And receptor-mediated endocytosis, which means that we're engulfing things, but in this case, they have receptors that are on the membrane that's going to recognize what needs to be engulfed so that it can go through endocytosis. There are receptors that are specific to those substances. These are just examples of phagocytosis here and receptor-mediated endocytosis, where we have the receptors that are on the cell membrane. They recognize specific substances that need to be engulfed, and then they are engulfed. Exocytosis. Exo means outside or out or exit. So it's leaving from the cell. So exocytosis, those things, for instance, we were talking about how things are made with the ribosomes or ER and transported via vesicles. Those vesicles are then going to fuse with the cell membrane, and release those contents. An example that we'll talk a lot about, or we'll talk in several parts of A&P, will be neurotransmitters that are being released. And so once they are made within our nerve cells, they're then transported to the cell membrane and then released whenever a signal needs to be sent to another nerve or another gland or muscle cell. And then we have transcytosis. Transcytosis means to change or to go from one place to the next. And so it is going to be receptor-mediated endocytosis. So it's receptor-mediated endocytosis. So it's endocytosis, but immediately followed by exocytosis. So something is first engulfed and then is going to exit. So endocytosis followed by exocytosis. Okay, so the next section is going to be on the cell cycle. Cell cycle, when we think of that, a lot of times we only think about mitosis, but the cell cycle is everything that happens before and after mitosis. And so we include interphase, which, although sometimes not really thought about when we think about the cell cycle, it's going to be the longest phase. Then we have mitosis, which is prophase, metaphase, anaphase. And then cytokinesis, which is the splitting of the cell. So again, interphase. The longest part of the cell cycle. What's happening in here, we have our S phase, G1 and G2. The cell is going to be doing a lot of growing. It's going to be creating a lot more DNA. It's going to be preparing for the cell to divide. So the reason why we have to have this is so that we can have enough DNA. We can have enough organelles so that when this one cell splits into two, we'll have enough organelles. We'll have a double of the amount of DNA. So we'll have everything that's necessary so that when it does split into two, everything will be fine. So we have G1, again, is growth. S phase is next. And then our G2 phase. So the phases in order, G1, S, and G2. So again, G1 is first. Then we have our S phase and our G2 phase. G1 is growth. Cells going to be growing, making more cells and more structures. Then S is for synthesis. We're synthesizing more DNA. And then our G2 phase, where the cell grows even more, preparing for our next phase, which is mitosis. And mitosis is simply going to be the splitting of the cell into two. From the parent cell, we'll eventually have two daughter cells. And so the phases of mitosis, you probably have heard of the acronym PMAT, so that you can remember them in order: prophase, metaphase, anaphase, and telophase. Very briefly, prophase is going to be where chromosomes are going to condense and form chromosomes. Those centrioles are going to, or the centrosome, where we have two centrioles, are going to go into the opposite ends of the cell. The nuclear envelope will then begin to kind of break down so that things can move around the way they need to. Meta means middle. This is going to be where the chromosomes are going to align in the middle of the cell between the centrioles. Those spindle fibers are going to form from those centrioles. Anaphase is going to be where the chromosomes are going to be separated because they have attached to the spindle fibers. They're going to be pulled away towards those centrioles. And then telophase is going to be where the two nuclei can then be visible, it will start to form. You can kind of see two cells forming, and those chromosomes are start to look more like chromatin again, and the nuclear envelopes will form around the two new nuclei. So again, we have prophase or early interphase here, or late interphase here, where it's still chromatin or centrioles. And then beginning prophase here, where the centrioles are moving to opposite ends, chromosomes are beginning to form. Late prophase, chromosomes are starting to form a little bit more, centrioles are on opposite ends. There are metaphase, where chromosomes are in the middle. Spindle fibers begin to attach. Anaphase, where the chromosomes are pulled to opposite ends. And then we have our telophase, where you see the nuclear envelope begins to form around both. And then eventually we have cytokinesis, where they're split into two. Again, cytokinesis is going to be right after telophase. We're at telophase. We are developing or seeing those two cells, and but it will begin early in anaphase, but it will finish right after telophase, where you have the cytoplasm that pinches in half and form your two new daughter cells. So we have to have some things that are going to kind of control this whole mitotic division and cell reproduction and replication. We don't want things to get too out of hand. And different cells can replicate a lot faster and a lot readily than others. Whereas skin cells, if you injure yourself on the skin, in a day or 2, 3 days, it'll go ahead and heal. In about 2 weeks, you may not even realize you even had a scratch, depending on how deep the scratch is. However, neurons, on the other hand, they don't have that long of a life where they can constantly divide. Early in life, they do divide and make more neurons. That's why brain development is so critical as a fetus and very, very young babies, because the brain is still growing and making more neurons. But after a while, it kind of ceases. And that's why it's so critical when a person damages neurons, whether it's the brain or neurons that are motor neurons or sensory neurons in your arms, legs, wherever. When they're damaged, it is very difficult for them, depending on the type of damage, for them to regain their ability to function properly. And so, again, all types of cells are going to be different. You have hormones that play a factor, growth factors, a lot of different things. And then even contact and inhibition. Inhibition means that something is stopped. To inhibit something means we're stopping something. Contact means that it's touching. And so if we have these healthy cells, they're dividing, they may stop dividing when it becomes too crowded, meaning that we have cells that are a little bit too tightly compact. We may say, okay, we need to stop dividing before tumors start to develop, because tumors are just overproduction of cells that's just kind of unregulated, and that's basically a tumor, because there's no control over mitosis. Those cells just continue to divide and go against contact inhibition. So we have two different types of tumors. You all probably have heard of both of these just in everyday language. We have benign and malignant. Benign tumors are those that are not cancerous. They are in this one area. They may not even get too much bigger. They could, but they're not going to get a lot bigger. But they're not going to develop in the lungs and then start to develop a little bit more in the breast or continue to develop in the pancreas. Benign tumor would not do that, but a malignant tumor will. And those are the ones that you will have to usually get treated. There people, some of you may even have had a benign tumor, and a benign tumor you can simply get removed, no problem. A doctor may want to make sure they just keep an eye on it, but if it's benign, then it's fine. And you can just get a regular test to see if a tumor is benign or not. And we have two different types of things within our body. In addition to contact inhibition, we have two different types of genes that are going to be responsible for causing cancer if they don't operate correctly. For instance, oncogenes. These are going to be abnormal forms of genes that are going to be responsible for controlling the cell cycle, but sometimes they may be overexpressed. And so if they're overexpressed, then we'll have that constant mitosis that's constantly happening, and they're abnormal, and so they could cause a tumor. And then we have tumor suppressor genes. Now, suppressor genes mean that it's going to slow down or hold back to suppress something. But if we're talking about tumor suppressor genes, that's what it's supposed to do. But if there's some type of issue with that gene, it becomes removed, inactivated. If there's some type of mutation, then we don't have a tumor suppressor gene that's functioning properly and could cause cancers or tumors. And so, one of our last sections is about different types of cells that we have. We already talked about how cells are differentiated, which means that they have different functions, shapes, locations. They're different. But they weren't always that way. Initially, we have a bunch of stem cells, and those stem cells are cells that can differentiate into a lot of different cells. They could turn into a muscle cell or a neuron or a skin cell. A progenitor cell is one that is almost committed to what it wants to be. Okay? So a progenitor cell we know that it's going to either be a muscle cell. We know it's going to be a nerve cell. So a progenitor cell would be a white blood cell, but we don't know which type of white blood cell. Or we know that it's going to be a muscle cell, but we don't know what type of muscle cell. And we have types of progenitor cells. A totipotent cell is also, again, that one that is a type of progenitor cell that can become any type, where it may be a fertilized egg, where it can go into any type of cell. It's more of a stem cell. It hasn't really decided what it wants to be. It's not committed. Pluripotent is limited. These are going to be your progenitor cells. And I misspoke when I said that totipotent cells are types of progenitor cells. Totipotent is more of a stem cell, whereas pluripotent is a progenitor cell. Progenitor cells and pluripotent cells are those that we know it's going to be a muscle cell, but we don't know what type of muscle cell. Or we know it's going to be a skin cell, but we don't know what type of skin cell. And this is just an example of that type of differentiation, where those stem cells can be any type of cell. It can be another stem cell, or it can turn into a progenitor cell, and that progenitor cell can be one that is committed, but committed to a certain, not specified what type of cell in that category. For instance, the example here, a myeloid progenitor cell. Myeloid is in reference to a white blood cell. But we have various types, so this progenitor cell could turn into a basophil or eosinophil or neutrophil or any of those because it is a progenitor cell. And after it gets to this point, it's a specialized cell and it won't differentiate into anything else. And our very last section is on cell death. We have two different types. The first is apoptosis, which is programmed cell death. This is cell death that happens all the time. It's supposed to happen. For instance, in the womb, we have webbed fingers. When we're born, we don't. In order to get rid of that, there has to be programmed cell death to remove the webbing between our fingers, or to remove the post-anal tail that we have at 5 to 6 weeks. Necrosis, however, is from damage. For instance, if I damage a finger and it's not able to get a blood supply, and those cells die and eventually maybe even fall off, that's necrosis. Okay, and that is the last slide for this chapter.